Here we go. And you can see the laser pointer, Josh, right?
That's so I can see it on our screen, but not on the. So I log in myself as just the.
Okay, I didn't do that, yeah, because of the delay.
Yeah, it's not on there yet.
Okay, we're
It's
on.
just it's nothing is happening on the connecting room inside 30 seconds.
Do I have? But you can see my screen.
Yes. So our presenter screen look fine. The as what do I call attendee screen now it's changing. So yes, I can see it both.
Okay. And I switch the slide, and it works. We have 13 attendees. Great. We're going to wait for just a minute or two to make sure those who are already signed in that we will start once we get a quorum, so to speak, or several more people. So it'll be a few minutes. Okay. Why don't I start? My name is Bob McCarthy from VitaCyte. Josh Sakon from the University of Arkansas will join me on this webinar presentation. Josh is one of the first groups that published on the three-dimensional structure of Clostridium histolyticum Collagenases. And of course, the purpose of his research is to try to define how these enzymes work to degrade collagen. So the webinar we're having today is Collagen Degradation by Clostridium histolyticum Collagenases, Importance, Measurement, and Mechanism for Cell Isolation. So without any further ado, we'll get started. The learning objectives for this webinar is to relate why this enzyme is important for successful human islet isolation. Its relevance, we'll describe the relevance of collagenase structure function to its performance and islet recovery. We'll have one slide just sorting through all the different collagenase assays that you're probably familiar with and show why the collagenase degradation activity assay, or referred to as CDA, is important to assess potency. I'll review two of the main assays that's being used today by commercial manufacturers. The Worthington, the Mandl assay that's used by Worthington and Sigma, that was developed in 1953, and an assay we developed here at VitaCyte in 2008, a fluorescent microtiter plate assay. Then Josh will cover the protein structure of class 1 and class 2 collagenase. These are two isoforms of this enzyme that behave differently in cutting collagen, and he'll also cover a hypothetical model of how collagen is degraded by these two isoforms and its similarity to cellulase digesting cellulose. So where do we begin? Well, we begin at collagenase P. And why collagenase P is important is Camilla Ricordi evaluated this product soon after its introduction in 1990 by Boehringer Mannheim Biochemicals. That's where I worked at the time. And he found it was the best enzyme for human islet isolation. His influence actually led to the development of the Liberase project. The success of any protein purification project, which of course the goal was to define what are the enzymes responsible for islet isolation, is to use the appropriate characterizations. And what we adopted right away is analytical anion exchange chromatography. And you can see on the left side a analytical anion exchange chromatography profile. For collagenase P, you can see these big peaks, collagenase 1 and 2, which are class 1 and class 2 that I mentioned earlier. We now know this is intact enzyme. These different forms here, the 2B, 1D, and so forth, are degraded forms of this enzyme. This is something we found out later. You also see in this crude collagenase a neutral protease, the C histolyticum neutral protease, and you have pigment and debris. And the reason is this is just a supernatant from fermentation broth. Since only about 30% of these lots were effective, we then used that fact to figure out what the common characteristics of good lots of collagenase were. And we looked at the literature and we determined that, hey, all you need is the 2 forms of collagenase and at least 1 protease. And in fact, this is the original Liberase HI product. As you can see, it's very clean, intact collagenase. And we couldn't use the C histolyticum neutral protease because it was unstable after purification, so we used thermolysin in its place. Everything went well with the Liberase HI from about 1995 to the early 2000s. And at that time, we had transferred the methodology to do the analytical anti-exchange chromatography to John Lakey when he was in Edmonton. And they started seeing a drop in their islet yields. And so as Matt Barnett in John's lab started doing these analyses, he found that this back shoulder was there, which indicated proteolyzed class 1 collagenase. And on top of this, the Clinical Islet Transplantation Consortium trial was started around 2004, and they were trying to look for a stable source of enzyme, and NIH didn't like the idea that this had a risk of potential transmission of TSE, transmissible spongiform encephalopathy, so they discontinued the Liberase use in all NIH-funded clinical trials, and they moved over to the Nordmark, Servin Nordmark NB1 collagenase product. The problem with that product that they found out soon is the leading centers who were very successful in repeating the Edmonton results couldn't get the same islet yields with the Servin Nordmark product. So in 2007, we did an analysis, and here you can see the intact C1 is even at lower levels than the other 2 forms. Again, correlating the form of this class 1 collagenase with its effectiveness and islet yield. And basically, we started using our own methodologies, our VitaCyte collagenase HA, and we're able to isolate an intact class 1 collagenase. We did further studies with the University of Minnesota. This was a paper published in 2010, and this is only a blow-up of the class 1 collagenase analytical anion exchange profile, but you can see the same observations. And here we have slight degradation of the C1, but it's primarily an intact C1 peak. The important thing to note is, which I'll get into this very shortly, is this collagen degradation activity measurement. It's a specific activity where you measure units per milligram. It was about 3-fold higher in the HA product. And this assay, which I'll describe shortly, was a fluorescent microtiter plate assay. It has some unique characteristics. Now, how's this tied to what's happening? Well, in the same paper, Dr. Bala Morgan showed that the digest time was shorter, and you had higher yields, which were all significantly higher than what you found in the Nordmark product. So how does this relate to collagenase structure function? I'm only going to touch on this because you'll hear more about this from Josh. Basically, in 1998, the studies were published out of Japan that sequenced these two isoforms, the class 1 and class 2, and in expressing the recombinant proteins, they showed it was a multi-domain protein structure, and it had 3 different domains: a catalytic domain, a PKD-like domain. Initially this was referred to as a linking domain because they didn't know the function, but now we know more about it. And it's a protein that has homology to the polycystic kidney disease-like protein. So we call it the PKD-like domain. And of course, the collagen-binding domains, which by definition, these independent domains bind to collagen. What the interesting observation was from the study was that functional collagenase requires both the collagen-binding domain and the catalytic domain to be on the same molecule to cleave native collagen. If you remove this collagen-binding domain, it won't effectively degrade native collagen. We did a study confirming this where we took one of our purified preparations of collagenase, and in fact, this was an early preparation where it was illustrative, and we took peak fractions from each of those peaks, and on portion of it we did collagen degradation activity assay and measured that activity, and the other we did SDS, polyacrylamide gel electrophoresis. And sure enough, what we found very interesting is the intact C1 with 2 collagen binding domains had a very high CDA activity, 100,000 CDA units per milligram, whereas the ones with 1 collagen binding domain, the C2, the C1b, or the C1c, had about a 10-fold lower concentration or activity, specific activity for collagen degrading activity. We call these forms now truncated C1 rather than degraded, because the truncated means that they're still active. They still have that, they still have a collagen binding domain on the enzyme structure. Of course, if you lose these domains theoretically, and we've shown this, that you don't have any activity. So how does this relate to correlation of collagenase structure to function? You need to have these collagen binding domains present to have collagen degrading activity. As all of you know, in the Islet world, we use the Wunsch assay, the PZ peptide, to measure it. It measures primarily C2. And unfortunately, if you lose a collagen binding domain, that activity stays. So it doesn't reflect the ability of the enzyme to degrade native collagen. And then that was shown in an earlier paper that Tatsuya Kin published in Edmonton. Now, how does this relate to what was done at Minnesota? And quickly, I can say that you have a high percent of truncated C1 is going to give you lower human islet yields than one with a higher amount of intact C1. These studies were like, it was 88 islet isolations performed at the University of Minnesota, and the report was published in 2012. The other thing they found out was interesting in this paper is the selection and dose of the neutral protease was very important. And they developed what was termed the new enzyme mixture, which is referred to an enzyme combination here. It's still being used at the University of Minnesota, and it contains the collagenase HA that we manufacture and the NB protease that Nordmark manufactures. And then the reason they use this combination is the NB protease was gentler on the islets and more effective than the thermolysin. So we incorporated this into a model, and there's 5 postulates for this model of enzyme-mediated cell isolation. And what we believe, and this is all just ideas of what we think is happening, is we know the extracellular matrix is resistant to proteolysis, with the collagen fibrils and fibers providing the superstructure to protect this matrix. We know that the purified C histolyticum collagenase is a very specific enzyme, degrading only gelatin and native collagen in the extracellular matrix. I've gone through the importance of having collagenase having a catalytic domain and at least one collagen-binding domain. We think what's happening, and this is why collagenase is so critical, is it has to loosen up the extracellular matrix, exposing protease-sensitive sites on other ECM proteins. And basically, once a sufficient number of these cell-anchoring proteins are cut, then the cells are released from the tissue. That's our working hypothesis, what's going on. Now, getting into the state-of-the-art of collagen degradation activity assays, the Mandl assay was developed in 1953, and it uses a collagen fiber substrate. It was really developed because they were looking at collagenase in wound debridement. And all you're mixing is the Achilles tendon fiber with the appropriate dilution of the enzyme, and incubate 2 to 5 hours. You then spin the undigested fiber down and take the portion of the supinate and measure free amino groups, and you calculate a specific activity based on the release of these amino groups. We've worked on developing this assay at VitaCyte, and basically in developing any assay, you want to have set up a condition where you have pseudo first order kinetics, which means everything is in excess except the enzyme that you're analyzing. So what we did is an experiment looking at a standard conditions of using collagen fibers, and you can see that this assay is pretty linear at 1 to 2 hours, but then it falls off the curve. It gets into a second order curve, which is nonlinear. Now, why is that important? Well, it depends on when you harvest and measure this activity. You'll get different values, and you can see here the values are the same at 1 and 2 hours, but you go out like to 5 hours, which is normally performed in the traditional Mandl assay, and you're going to get variable results. And what we think what's happening here is that as the collagenase is attacking the collagen fibers, there's easy sites to cut, and then it gets more difficult sites to cut. And what you're trying to do is try to get a reproducible measurement of this activity, and increasing the incubation time causes problems. The other assay that we developed in 2008 was a fluorescent kinetic microtiter plate assay. It's a homogeneous assay. You don't need any separation. You just put the collagenase in with the FITC fibrils and incubate for an hour. And the beauty with the microtiter plate reader is you can take multiple measurements looking at the values over time. What we do in a representative result is shown on the next slide is we actually measure always in duplicate. We measure always at 3 different dilutions, and we take the mean of 7 points off this curve. And of course, this is in pseudo first order kinetics. It's linear, and you can see the calculation of multiple dilutions of the same sample. We get a value, and we get a respectable inter-run CV at 7.3%. So the comparison of the CDA assays. The Mandl assay, which I mentioned before, You're measuring free amine groups. When we've done studies with intact C1, which is the IC1, and truncated C1, the TC1, what we find it very interesting is they're about the same specific activities, whereas C2 is about fourfold lower, I should say. The problem is it's an ill-defined substrate. The specificity is dependent on removal of telepeptides, which I don't know whether these substrates have that done, and the precision is not reported. The fluorescent assay we've been using since 2008 and improved it over time. And basically, we see differences in each molecular form. The intact C1 has a higher specific activity than the truncated C1, which is higher than the intact C2, as shown in these graphs below. The beauty of this assay is that it's a specific CDA assay. If you add excess protease, you get very minimal digestion of the substrate, and the precision is good. And we can talk about this later. We're evaluating the transferability of the standard. So the question really gets into is how come these two isoforms, these truncated form and the intact form, are about the same where it's different here? Well, we think is it gets into the assay mechanism for doing a fluorescent enzyme assay. And what happens when the FITC is in close proximity to other residues within the protein, and it's a folded protein, they're close together, you get self-quenching. And the emitted fluorescence is suppressed. In other words, it gets absorbed by neighboring FITC or other amino acid residues. When you cut with the enzyme, you're actually freeing it from that environment. And now when you excite that fluorophore, it does emit the fluorescence. And this was a schematic that was done for an assay that was for a protease assay. And basically, we think it's even a little more refined with using a collagen degradation activity assay. In the fact, we think that what's happening is you unwind the protein, you're actually seeing increases in fluorescence. So at this time, I'm going to hand it over to Josh to talk about the structure, and I'll turn off my share screen. Okay, Josh. Yeah, I'm just going to break in here. Josh, I think you may be muted. We can't hear you. Double check your...
Got it. Are you able to hear me?
Yes, I am now.
Okay.
Okay.
Thank you. So collagenase has a tall task. Type 1 collagen is a very difficult substrate. to degrade. The procollagen that you're seeing here is cross-linked to make a rope-like structure. They're stacked on top of each other in a regular fashion and is being twisted. This gap even shows up in the electron microscopy or the AFM. It is that regular of a structure. So for the enzyme to degrade this type 1 collagen, it first needs to unbundle this fibril to microfibril, which you are showing here on the right side. Each tropocollagen, that is a triple helix, is shown in a different color. The enzyme will have to swell this, meaning that it will not disrupt the triple helical structure, but the unbundle, so to speak, the term that we use is swell, to isolate each of those triple helical collagen, and then have to unwind this. Each one of those, let's say it's a red one, consists of 3 polypeptides. Each one is colored differently. So green is one polypeptide, the pink is another, and blue is another. They are tightly wound together to form this triple helix, and the structure hides the peptide to be cleaved, the peptide bond to be cleaved. So for enzyme to tackle this substrate, it will first have to unbundle, then swell and unwind the triple helix. In order to carry out this function, the bacterial collagenase consists of 3 domains that Bob talked about. The catalytic domain oftentimes is called the collagenase unit, but it has no collagenase activity. It is a gelatinase. It utilizes zinc to activate water molecule to hydrolyze the peptide bond. It has a PKD-like domain, and the role of this is to swell the collagen fibril. At least that's the current hypothesis. And then it has a collagen-binding domain that unidirectionally binds to collagen and prefers to bind under twisted triple helix. Those 3 domains, 3 units, are linked together by a polypeptide. And what you're seeing here in green is a calcium, and the linker and the domain itself binds to calcium to transform the structure. So and that I will show you in the next slide. So that the important thing here is that it has a zinc for the catalysis to take place and a calcium to rearrange the domain to activate this enzyme. It is showing here with data that is called its axis, but in the absence of the calcium or very low calcium, it adapts a very elongated structure, and the electron density that you see excess, it is because that it adapts ensemble of structures that will actually condense into the structure that is shorter in length and a more compact structure in the presence of calcium, so that the calcium is needed to what we consider this activated structure. Once you have activated the structure, it can degrade the enzymes in 2 different ways. So we call it Coll G, but this is the Bob's C1. And the Coll H is, in Bob's slide is C2. We just use a different nomenclature, and also that we use a different name for the species, but it's the same thing. The name changed from Clostridial to Hathawaya very recently, and we have to follow that. So that's the reason why the slide started with Hathaway. But Coll-G is C1. In the audience, Dr. Tanaka is there, who carried out this catalysis. What you're seeing here is an intact collagen that separate using SDS page. This band represents alpha 1, and this band represents alpha 2. Because that type 1 collagen is a heterotrimer, you see twice more intensity for the alpha 1 than to alpha 2. They are cross-linked so that it appears at the twice the molecule weight. And because that collagen is further cross-linked, you see even the 3 times the molecule weight size that shows up at the where I'm pointing. When you add the collagenase C1, I hope that you can see that intensity decrease over time. Intensity of all the bands decrease over time. And that is because that this is an enzyme that is not fragmenting the collagenase, but rather thinning the collagen. So we believe this enzyme is a processive protease, and in the next slide I will show you the high-speed AFM results that our competitor performed. Coll-H in it is in Bob's slide it was C2. This is the collagen, and again it has the alpha band, beta band, and gamma band. This is all these higher molecule weight bands show up because that collagen is cross-linked. The results after you adding the Coll-H is that it fragments in a specific way, as if it has a hypersensitive sites. It fragments further and further and further as the time goes. This is a characteristic of what we think it is for the endopeptidase, meaning that it will cleave somewhere in the middle of the collagen. And it's not really somewhere, but it has a specific sites that it prefers to cut. And those two enzymes work together to degrade the collagen, and that I'll demonstrate. I'll show you the results a little bit later. So they work synergistically. The Coll-G, or in your world is C1, is, I told you it's a processive enzyme, and this results demonstrate you that it is processive. What you're seeing here is the AFM of the collagen that is oriented C-terminus near the top and N-terminus below. One can monitor the movement of the Coll-G that is highlighted in green. I hope that you can see that the green dots move towards the N-terminus. The pace is 3 nanometer per second. And every path, the collagen is thinned. So that we think this is the operating mode for the Coll-G. It is a processive enzyme that act almost like a planer to thin collagen. In order to function as such, the Coll-G looks like this. In other words, it looks like this on the right side. It's led by collagen-binding domain that binds to collagen in unidirectional fashion, so it positions the rest of the enzyme to the right way. What you're seeing here is the second collagen binding domain, and what's here is actually the what we think it is a second collagen binding domain. So they position the enzyme so that PKD will follow. PKD, what we consider it as the function is to swell so that the tropocollagen can be fed into the catalytic segment. And let me show you this molecule from end on so that you'll be looking this molecule from the left. So you'll be looking at the arrow that I'm drawing. From the end, it looks like a Pac-Man, ready to accept the tropocollagen. And we think that the chewing action that of this two segment that is in the catalytic domain unwind the tropocollagen and hydrolyze the triple helix. So this molecule will move towards the right, and the and in a processive fashion, it will stay on the enzyme and the chew up the what will be the C terminus of the collagen, and it's so collagen will be sitting right here. In the pancreas, it consists of 1 to 6 type collagens, and with the presence of both Coll H and the Coll G that I show you, can degrade the type 1 collagen. Again, this is alphas, and that's the beta band. That is a cross-link, and that's gamma. It can degrade type 3, and type 4 is network type collagen, and it's degraded. And type 5 is also degraded, but not so much for type 6, which is abundant in the pancreas. You see the band remaining, and we think it is because of the second collagen binding domain. We determined the structure of the 2 collagen binding domain of the Coll G, or C1 in your world, and it binds to triple helix that are oriented in parallel. So C terminus of the collagen will be here, and the C terminus of collagen will be here. And let me hold on to the N terminus and swing it towards you. That will be the structure that you're seeing on the right. It has internal twofold axis. So this is a first CBD, and that's a second CBD of the Coll G. And it wedges between the parallelly oriented collagen molecules. In the type 6, the collagen molecules are oriented in antiparallel fashion, and the Coll G could still bind using one of the collagen binding domain to the substrate, but may not be able to process. And that might be the reason why it is not so active towards the type 6. Let me show you how those, what we think of how Coll G and Coll H work together to degrade collagen fibril. What you're seeing here is the Coll G and Coll H, and Coll G is a planar. The Coll H is more like a saw. When the Coll H cleaves, it will generate new C terminus where the Coll G can begin planing. So that could be one of the ways that Coll G and Coll H work together synergistically to degrade collagen fibril. The other thoughts that we have about the synergy is the way that collagen fibril is made, the position. So the top is showing you the triple helix. It's a collagen, it's 300 nanometers long, 1,000 amino acids, that adapted this triple helix. It is colored so that colored in 5 zones from D1, D2 to D5. The collagens are stacked on top of each other in staggered fashion, and let me focus on this region. So 5 triple collagen makes up microfibril, and let's focus on just this region that has all regions represented. So D5 on the top, D4, D3, D2, D1 are is a segment of the microfibril. In this 3 dimension, based on the fiber diffraction data that was completed by the Ogles lab, suggests that C5, the E5 region is on the surface of the fibril. So what you're seeing here is just the blow-up picture of the surface. So that would be this. D5 is on the top, and D4 is below, and 2 is below that, and 3 is in the interior. And finally, 1 is on the most interior of the fibril. The cutting site for the Coll H is hidden. The cutting site, the hypersensitive site for the Coll H is found on D2, that will be this, and they found on a D4. In order to access the cutting site, the D5 needs to be removed. Fortunately, that's something Coll-G can do. In this diagram, this will be the C terminus. So Coll-G can remove this D5 segment of the turbocollagen that will expose the 4 and 2 region where the Coll H can make its cut. There, the Coll-G can begin the planing action. So the collagenase will tackle from the outside and penetrate to the center of the fibril. So what I presented here are based on the incomplete data that we have, some structure, some functional, to try to relate the structure and function. Each domain in bacterial collagenase has a unique role to play, and they evolved to be able to degrade this highly ordered, difficult substrate. The approach to degrade resembles how the cellulase breaks down cellulose. Cellulose is the extracellular matrix for plants, and it is another formidable substrate. And the approach that bacterial collagenase evolved is similar. It has different enzymes with different functions to be able to efficiently degrade this insoluble and tough substrate. It requires calcium to reposition the domains. It requires zinc atom to activate water so that it can hydrolyze the peptide bond. The current hypothesis called G, based on is primary processive exo-like peptidase. The Coll H is primary endopeptidase. It will cut in the middle of the peptide. So I'll go back to Bob.
Okay, thank you, Josh, very much for the presentation. And as we all can see, it's kind of a tough nut to crack to find out how these work. I'm going to turn on the last slide. Let's see, from the current slide. Hopefully you can see that now. Share. Yeah. Oh, it's all right. Yeah. The implications for this new information. I think what Josh showed is reiterated the importance of calcium to collagenase structure and zinc to catalytic function. I know there's a lot of discussion about this. You want to avoid chelating agents. We've also found that people who stored their collagenase and phosphate buffered saline and froze it will lose activity because the phosphate is a chelating agent. And secondly, and this is a lot of discussion, and somebody may want to comment. When I've talked to Josh about this, 1 millimolar calcium should be sufficient to protect the enzyme. It's really important to realize that in low calcium, you're actually changing the structure and more than likely making the collagenase susceptible to proteolysis. The other issue that I think this comes up with is from Josh's talk is that these are processive enzymes at 35 to 37 degrees. They're active. However, at 4 degrees, they can stick very tightly and bind to cells. So you have to be careful not to overinterpret what you're seeing in collagenase binding to cells, because normally my guess would be is that it just falls off the cells and isn't as big a problem as we think it could be. And to put this in perspective, recently Endo Pharmaceuticals used collagenase, an injectable purified C histolyticum collagenase, the product's called Quo, to remove cellulite, cosmetic application. And the amazing thing about this is that nearly all the patients are developing anti-C1 and anti-C2 antibodies, and they're injecting a lot of collagenase. So again, this gets into this whole issue about the relevance of the residual collagenase, because in essence, the FDA has approved use of this product, and yet people are developing antibodies to it. So I don't know if we have any questions. I can start off with just an idea as you were talking, Josh. What we've found is that there's all different ratios of class 1 and class 2 that still work effectively to degrade native collagen and release cells from tissue. We've gone typically for most food collagenase, it's 75% class 1 and 25% class 2, but yet when Bala was using our recombinant, the mixture that he used and the lowest amount of enzyme that he used, just the way the experiment was designed, was 70% C2 and only 30% C1. So my question to you, would you expect to have different, and I don't know if this is, I'm sure the experiments that have been performed, but different profiles of the peptides released by these two extreme mixtures, primarily class 1, say 60-40 or 75-25 versus the other way around, where the C2 predominates. Do you think you'd see differences in the mass spec profiles of the peptides released?
I'm not so sure that we can actually see it experimentally.
Okay.
Yeah. When the smaller fragments are generated, if they're not cross-linked, they have such a low melting temperature that it will probably solubilize
Okay.
And those are the ones that mass spec are really good at observing, whereas the longer ones they can't, and the not as well. So yeah, I think we'll see different results. I'm not
Okay.
so sure that we can examine. Yeah, I think we'll be able to tell the different products being made.
And I think the point you're bringing up is you may not have to have complete digestion of in our case, we were looking for cell release. We may not probably have to have complete digestion of the collagen fibril or fiber to get the result of cell release based on your model, because it's going to disrupt that extracellular matrix.
Yeah, I think at least what I read suggests that oftentimes you have to add back extracellular matrix in order to sustain the cells from going apoptosis.
Yes.
So by partially hydrolyzing it, partially disrupting the extracellular matrix by collagenase is the way to go, not using some indiscriminate digestion by other enzymes.
Well, clearly, I mean, the way that people who isolate cells do is that's the issue. You don't have a fixed time point, and you have to look at what the tissue is actually doing before you call a reaction.
Yeah.
Because I think that gets into damaging the cells in many different ways.
And I think depending on how the pancreas is, how old it is, what is the disease state, cross-linking level probably differs. So cross-link the blocks expression might be different in the different person too. So may have to adjust the ratio of the C1 and C2 or Coll G and Coll H to optimize the release of the pancreatic islet. That would be my guess.
Okay.
Because substrate is so heterogeneous. My naive guess is that adding more endopeptidase, which will be C2, may be able to degrade highly cross-linked substrate better. But I could easily be wrong about that.
Okay, Drew Brady had a question for you, Josh. Are you able to comment on the role of the protease might play with the Coll G and Coll H to accelerate the degradation of the extracellular matrix?
Yeah, so I don't study those clostopane and the thermolysin myself, but there are relatively non-specific heat stable, in other words, they can tolerate the temperature ranges, and they are quite active enzymes, and they may be going after laminin, they may be going after fibrin, they will go after accessible, relatively disordered loop regions of the extracellular matrix. I think that's what they're doing.
See, what I was thinking, to add on to what Drew was saying, is that actually those proteases are also does it accelerate it?
Yeah, it can. It might be doing that too. So if the product is long enough, it might be redeposit on the collagen. And if the the thermolysin or clostiphanes around, they will hydrolyze them, so they will be small. And once it gets too small, there will be no way in the world that they will be redeposited on the collagen. And that's the strategy that cellulose degradation use. They use in the cellulose, they don't have the N terminus and C terminus, but they have the non-reducing end and reducing end. And there will be enzyme that comes from one end. They will have the endotype enzyme that will cut in the middle. But once the products are made, there will be another enzyme ready to dice them up. So the beta glucosidase will be ready to make it to sugar, glucose. So that could be what the role of the thermolysin is.
Dr. Swee from Georgetown said, do you consider the process is more than manipulation if you utilize the collagenase during the cell processing? Trying to make sure I understand what he's asking. I think he's getting more. This gets into, I'm assuming, and Dr. Swee can correct me, but I'm assuming this gets into this whole issue, I think, of what the FDA is saying is it's more than minimal manipulation. And this is a whole can of worms, because see, they allow in the United States preparation of islets for auto islet transplant is not having that there is, they allow it because there isn't minimal manipulation. Yet at the same time, if you're isolating stromal vascular fraction from fat, they say that's enzyme treatment is more than minimal manipulation. So they're kind of schizophrenic in how they interpret this. So, Rangting, if you want me to clarify anything else, just send me another text. One other thing I want to make note of is, we found this when we studied the collagenase and isolating islets, is very interesting. Is it appears that both pig islets and human islets require the combination to work in a complementary fashion. But there are two reports out, one done in '94 and one done probably several years ago, that show that actually C2, class 2, or the Coll H alone can isolate rodent islets, mouse islets.
Yeah.
And so the question really gets into, what does that mean in terms of, I assume we just have different structures. And that's why we typically find that there's different enzyme for each different tissue, and each same tissue, different species, you can find different compositions are optimized. They're not the same. So
yeah, I think I saw the results from dog, and they use a different combinations.
The dog, we developed one with the liberase, and it's very different than the human. Yeah, the dog has a very, very thick capsule. Well, our time is just about up for an hour. If there's any other comments or questions, we can reply to them, but by email. I hope you found this beneficial. It was a change of pace to get more into the details. I will say this: we are pursuing working with NIST to try to figure out how we can standardize how we, with different plate readers, people could start doing this assay. And it's something in the top of our mind. And we just had an initial call with the NIST people about this. But I think that this is an important assay that needs to be more available to all, to make sure that their enzymes are working appropriately. So thank you again for everything, and thank you, Josh, for this great discussion. And again, feel free to contact myself or Josh if you have any question. All right, thanks. Bye.